Senior Research Fellow at University of Lausanne, Switzerland. You may type in a question at any time during the webinar. You can do this through the Q&A panel, which appears on the right side of the webinar presentation. If you look to the bottom tray of your window, there are a series of widgets to enhance your webinar experience. And with that, I'll turn it over to Spencer.
Great. Thanks so much. So yeah, thank you again to 10x Genomics for sponsoring today's webinar, and thanks everyone for tuning in today. So the topic of today's talk is using spatial multiomics, and in this case, using it to investigate the role that fibrosis plays in promoting glioblastoma recurrence. So a quick bit of background on glioblastoma. I'm sure many people are aware. This is the most common and most devastating tumor type that arises from the central nervous system. And the standard of care treatment for especially high-grade glioblastoma is typically surgical resection followed by radiotherapy and temozolomide-based chemotherapy. But despite these treatments, the median survival time for patients is still less than 14 months from the time of initial diagnosis. And what makes this such a challenging disease to treat is this huge amount of plasticity and heterogeneity that exists within the tumor cell population. And that's really going to complicate any treatment that's trying to specifically target the tumor cells alone. So that's why a lot of the field is now moving towards trying to target the more genomically stable tumor microenvironment, in particular, the immune cell population. So we have lots of clinical trials going now targeting T cells and dendritic cells. And one of the cell types that we focused on in the lab of Professor Joanna Joyce was tumor-associated macrophages, and using anti-CSF1R inhibitors, such as this agent BLZ945, to try to re-educate TAMs from a pro-tumoral phenotype back into an anti-tumoral phenotype. And in mouse models of glioblastoma, this was very successful. So we were able to shrink down even very large tumors down to small dormant lesions. And if you look at the volume curve here at the top, you can see that we get really rapid regression in just 7 to 14 days down to a period of dormancy that we define as being around 28 days following treatment. But after this dormancy phase, we did see that about 44% of tumors did rebound under treatment. But what was really interesting about these is that every time we saw tumor recurrence, 100% of these rebounds, they always regrew next to a glial scar that had formed during the treatment. So this led us to the hypothesis that these glial scars weren't just some byproduct of the treatment, that they were actually playing a pro-tumoral role in promoting recurrence. And then it follows, if we could inhibit the formation of these scars, then we could potentially improve the response to the immunotherapy. So just a tiny bit of background on glial scarring. So this is a bit different than scarring in the skin or other epithelial tissues. So following mechanical damage or oxidative stress or any like severe tissue insult, you get waves of cellular responses that eventually result in a multi-compartmental type of scar. So surrounding the scar, you get what's called the glial part of the scar. And that's where you have these hypertrophic reactive astrocytes. They increase in size and number, and they link up together, and they form this cellular border around the site of damage. But if the insult is severe enough, you get a fibrotic core in the center of the scar. And so this spatial compartmentalization becomes very important when we're trying to assess its role in the tumor microenvironment. So one of the models we use for this is called the RCAS model. So this is a viral-driven model that drives tumor growth with overexpression of PDGFB. But because it's a viral transfection, we can also do other modifications. So in this case, we've added GFP to the tumor cell population so we could see where they go after treatment. So looking at the MRI volumes on the left here, we can see that following the initiation of treatment, when the tumors shrink down and there's glial scarring, what's left is this large, dark, contiguous area in the MRI signal. And I'll point out that it's immediately adjacent to these that we see recurrent growth upon rebound. So we did correlative IF to try to determine what these structures were made of, and found that they were made of really dense deposition of ECM proteins. So in particular, collagen, fibronectin, tenacin C. And these were bordered by, again, these hypertrophic reactive astrocytes. And really importantly, we didn't only see this scarring response to treatment following anti-CSF1R treatment, we also saw it following more standard models of treatment, such as radiotherapy and surgical tumor resection. And we also saw in other models, so not just the RCAS model, but also in this HRAS GFAB CRE model, which is interesting because this is also in a different anatomic location. So it seems to be independent of location or treatment type. The idea is that any treatment that's aggressive enough to perturb the microenvironment has the potential to trigger this wound healing glial scar reaction. So to try to validate this in the patient setting, we teamed up with some neurosurgeons at CHUV University Hospital in Lausanne. And so when patients would come in for surgery for their recurrent disease, we had the opportunity to sit down with their MRI and identify regions of putative scarring that was associated with the treatment for the primary tumor. And then when the patient went into the surgery theater, we could specifically get those regions. And this allowed us to look at the interplay between the recurrent tumor and the treatment-associated fibrosis and see that it was very similar to what we saw in the mouse model. So we wanted to try to investigate how these scars were forming and what they were comprised of in terms of cells and protein. And so to try to get at some of the temporal nature of this, we used several different omic approaches on various different time points. So the key in the bottom here shows which time points we used each of these modalities on. So what we first did was single-cell RNA-seq on untreated samples and samples from 7 days and 14 days of treatment. And we next did a very large-scale mass spec proteomics study where we also did manual microdissection to isolate individual regions from within each section so that we could pool these together across each of the time points and then do mass spec-based proteomics. And this was very useful for us because if we were doing, like, say, GSEA analysis and aggregating and clustering all these different enriched pathways, we could compare not just between different time points, but between different regions of the same time point. And while both of these were useful, they are still largely just disaggregated data. So they really lack the single cell spatial resolution that we need. So we turned to a couple spatial omic approaches to complement these. And the first one was a spatial proteomic approach. So this is a variant of cyclic immunofluorescence called hyperplexed immunofluorescent imaging, or HiFi. And this was developed specifically for this project and includes a digital pathology workflow to analyze the data that uses machine learning to identify different structural features in the tumor. So obviously for us, we're interested in fibrosis, but also vessels, the perivascular space, regions of necrosis, or the leading edge. So that when we identify the individual cell types, we can look at where they are in relation to each other, but we can also look at where they are in relation to some of these larger macro features in the microenvironment. And so to add a spatial resolution to gene expression, we turned to the Xenium in situ platform. And here we used the mouse brain gene panel from 10x and then supplemented it with 100 extra genes to try to target some of the cell types and pathways that we're interested in the tumor microenvironment. So I think if anyone's used any of these, they know that on their own, these are all very powerful, very useful approaches. But the real strength for us was in integrating them together. And we had a couple different strategies for that. The first was what we'd call pre-integration, where the results of one experiment would inform the design of another so that it's not just completely done in the dark. And then after that, we have post-integration, where we're actually combining the resulting data to create a single multimodal data set. So I'll go through just a few examples of some of the integrations that we did. So between proteomics and spatial proteomics, obviously the proteomics was essential in forming the antibody panel that we would use for the high dimensional imaging. And this was also useful for validating a lot of the proteins that we identified, especially some of the ECM proteins in the proteomics. And between the single-cell RNA-seq and the high-dimensional imaging, so again, the single-cell RNA-seq was very critical in informing the antibody panel, because this would tell us what cell types we should really be focusing on. But it also works in the other direction, where we can take the cell-cell nearest neighbor distances from the digital pathology and use that to inform cell communication analysis on the disaggregated single-cell RNA-seq data. And I will go into that a bit more later. So another very useful one for us was combining spatial proteomics and transcriptomics. And we did this a couple different ways. And one was physically. So we would actually, after the Xenium run, we would take those same slides, perform hi-fi on them, do the digital pathology analysis, and then combine them so that we had on one image, we had gene expression and protein expression. And then we could also take a lot of these machine learning based structural annotations, add that to the spatial transcriptomics so that we could look at how gene expression correlated to proximity to some of these different structures. And then finally, and I think maybe the most powerful out of all of these, was combining the disaggregated single-cell RNA-seq data and spatial transcriptomics. So first, the single-cell data was very useful in figuring out what additional genes we should add to the gene panel, but it also allows us to project essentially whole exome data onto comparable cell types in the spatial transcriptomics. And because we had this sort of proximity to different structures, different cellular neighborhoods in the transcriptomic data, we could generate a spatial signature that we could then project back onto the disaggregated single cell data. So this was very useful for a number of different analyses in this project. So I want to go through some of what we discovered in analyzing this spatial multiomic data set. So first, looking at where we could find surviving tumor cells after treatment, we found that we could only find them in the fibrotic part of the glial scar, particularly in areas that were high in tenascent C. And using the Xenium analysis, we were able to find that all the cells within this fibrotic niche were highly enriched in a dormant stem-like gene signature. And this has clear clinical implications because cells that have dormant stem-like phenotypes are going to be resistant to additional treatments that are targeting neoplastic cells like radiotherapy and chemotherapy. So now looking at some of the immune populations, and obviously our major focus here are TAMs. So in this glioblastoma model, we have a huge amount of TAMs in the untreated context. But following treatment, we found that these macrophages, again, these are the macrophages that we're targeting with this anti-CSF1R treatment, we find that they're excluded from these regions where the surviving tumor cells are. And using proximity data from the hi-fi analysis, we can see that over time, this sort of disassociation of these two cell types increases over time until we hit this dormancy period around 28 days, where these cells are now almost completely mutually exclusive. And we have a different story with the T cells. So in this model, we don't get a lot of T cell innervation with in the untreated context, but following treatment, especially at this point of dormancy, we have a huge influx of T cells specifically into these regions of fibrosis. So the T cells are now in very close association with the surviving tumor cells. But with looking at Project TIL's analysis at the T cells and the single cell RNA-seq data, especially those 7 days and 14 days of treatment, we found that all the T cells, specifically the ones in this fibrotic niche, had exhausted phenotypes. So I guess sort of combining these last three slides, it sort of paints the picture of the fibrotic part of the glial scar as this protective niche that's holding tumor cells in a state of dormancy. It's protecting them from immune surveillance, from macrophages and T cells, until conditions are optimal that they can then escape from these structures and then regrow back out into the brain. So we wanted to validate this idea that these dormant tumor cells were the cellular source of recurrence at the point of rebound. And for that, we turned to Xenium analysis of rebound samples. And what we did was we took the tumor cell population, we binned them into concentric circles moving away from the point of fibrosis that formed during treatment. And we took the gene expression of the tumor cells in each of these bins and compared them to tumor cells at the 28-day dormant phase. And what we found is that, which button here? This one. Is that the nearest two concentric rings especially had very similar gene signatures compared to the dormancy genes, particularly in some of these dormancy-related genes. And this similarity to the dormant cells became less and less as you move away from the fibrotic scar, which again sort of paints the picture of the cells surviving in this fibrotic niche, escaping out into the environment, regaining proliferative capacity, and then further modifying as they grow back out into the brain parenchyma. So one of the questions actually we never quite answered in this project was how these cells are escaping the scars. So it could be that the ECM is being digested by the environment. It could just be through stochastic migration out of and just sort of finding their way out by random. But either way, when they do escape out into the brain, they find themselves in a highly growth-promoting environment. So using proteomic analysis, we were able to find that when comparing these different regions, the scar, the rebound tumor, the healthy brain, the scar was very enriched in a number of different mitogenic paracrine growth factors. So that once these cells are out into the brain, they're in a pool of wound-healing growth factors that can drive really explosive growth, which, looking back at some of these volumes from rebound, we can see very aggressive growth in these rebound samples, which can be much, much faster than how they grew initially. So, but okay, going back to this, just want to say, so this is now saying that the fibrotic scar is not just a survival promoting niche, but upon recurrence, it is also potentiating and promoting even more aggressive growth at the point of rebound for these tumors. Okay, so we next wanted to try to identify, okay, we're focusing now on the fibrotic part of the scar. What are the cell types that are most key and responsible for creating the fibrotic part of the niche? And we focused in on this group of cells that were transcriptionally similar to pericytes, but we are just calling perivascular-derived fibroblast-like cells. And one cluster of these in particular, this one here on the right, these had the highest expression of any of the cells in the data for collagen 1, fibronectin, all of the ECM proteins, and they were very uniquely associated with post-treatment samples. So we really didn't see this cellular phenotype in the untreated context. And so we used the proteomic data to create a gene signature for fibrosis in particular, and we found that this PDFL cluster had the highest unique expression of this entire gene signature. So the next thing that we want to try to figure out was, what were the signals following treatment that were driving these cells to produce all of these different ECM proteins? And for that, we turned to NicheNet-based cell communication analysis. And for anyone that's done cell communication analysis with any of the available databases on disaggregated data, you find that you get lots, you get tons and tons of different cell communication events that are very significant in terms of the data. So it becomes very difficult to try to assess which are the ones that are the most biologically relevant. So what we did was to use the cell-cell distances from the HiFi data and created probabilities of cells being within 30 to 50 microns of each other, and then used those probabilities to weigh and filter the NicheNet cell communication data to try to find those that were the most biologically relevant because they were the most likely to be happening in the TME. So we focused in on these PDFL cells. This is the blue line up top, the blue receiver cells. And what we found is that several different cell types were producing a range of different TGF betas and inflammatory proteins such as IL-1B. And this was driving very specific expression of a lot of the ECM proteins from that fibrotic gene signature. So we wanted to validate this by using the combined spatial transcriptomic and proteomic data. And so here we're looking at ERTR7, that's in magenta. That's a marker of putative fibroblast-like cells in the CNS. And with the xenium data, we looked for cells that had the highest co-expression of both the TGF-beta receptor and the downstream collagen-1 expression, and then found that there was a very high, almost perfect association between the cells that were the highest co-expressing of these and these PDFL cells. And these were interesting for a number of reasons, and one of them was the time dynamics of these cells. So in the untreated context, we really didn't find many of this cell type in the environment. We saw some like associated with very large vessels, but almost immediately after treatment, we get sort of explosive growth of this cell population, even just one day after treatment, so that by day 5 or day 7, these cells have spread out through the entire tumor and encapsulated the entire structure in fibrotic ECM. And this is very similar to this idea that's been out for a couple of years now of these type A pericytes. So this is a type of pericyte that exists along the vasculature in the CNS. And then following any sort of tissue insult, they delaminate, they detach from the vasculature, undergo waves of proliferation and differentiation to become fibroblast-like cells to stage a wound healing response in the CNS. And so this looks very much like what we're seeing here in the post-treatment glioblastoma environment. So we looked at some GSEA analysis. So now looking at 7 days versus untreated, and we see a lot of pathways that would be very related to this idea of type A pericytes. So we see EMT pathways, ECM organization, we see response to interferon gamma and alpha. But what was interesting was when we looked at a later time point, when we compared 14 days of treatment to 7 days of treatment, we saw that a lot of these same pathways now became downregulated. So we made some gene signatures based on these different pathways for EMT, ECM, interferon gamma, and alpha, and tracked them over time. And what we saw is that from untreated to 7 days, we get this huge increase in expression of all of these gene signatures. But after that, they all either plateau or spontaneously abrogate on their own. So in sake of time, there's a lot of different things that's very interesting about this, but for us, the clinical implication was the most important here, because what this tells us is that if we want to try to interfere, if we want to inhibit these cells' response to treatment, we have a limited window of intervention that we can affect any change, because since these pathways are going down on their own, it's very unlikely that treating post 10 days is going to do anything to the cellular response. So we really need to try to target these PDFL populations at the earliest possible time point. And so we devised a treatment strategy using a combination therapy targeting neuroinflammation and TGF-beta. So we had tested many, many different single agents over several years, and none of them were successful as single agents. It was only when we took this combination approach that we were able to affect any change. So to target neuroinflammation, we used a very broad inhibitor. We used dexamethasone at just for the first 2 weeks and then tapering off. And then for TGF-beta, we used a very specific inhibitor, galenisterib, and targeted that to just the first 10 days of anti-CSF1R treatment. And so this is useful in 2 ways. First is that we're targeting just this window of intervention for the PDFL cells, but this also gets around a lot of the really severe toxicity issues that are associated with using either of these agents long term. So now looking at just 14 days to do some validation work on this, you can see example images up the top, and at the bottom left, you can see a quantification of the amount of fibrosis. So we see that when we add dexamethasone to PLZ, this doesn't do anything on its own. This combination isn't sufficient to block fibrosis. When we add galenisterib, we do get some change, but it's not significant. We do still have fibrosis remaining. But when we add all 3 agents together, we almost completely eliminate fibrosis following treatment. And if you look at the graph to the right, this is the amount of tumor cells remaining after treatment. And you can see how highly associated the amount of tumor cells remaining are to the amount of fibrosis remaining. So based on this, we started some long-term preclinical trials, and then looking at progression-free survival of each of these agents alone and in combination. So the slide's a little messed up here, but the red line that you can hopefully see here is BLZ alone. And with that, we got about 50% survival, similar to previous studies. When we added dexamethasone and galenistrib on their own, without BLZ, we get no survival benefit whatsoever. It's as if we just give these mice vehicle, and sadly, they die off very quickly. So now in the orange line here, adding dexamethasone to BLZ, this does nothing in terms of survival. When we add galenistrib to BLZ, it maybe shifts the curve a little bit, but it's not significant. But when we add all three agents together, we go from about 50% survival to about 95% survival. In fact, we only lost one mouse in this arm of the trial. And so this really gets back to kind of our core hypothesis here, that this fibrotic response to treatment isn't just some benign byproduct of treatment. We can see now that it's playing multiple roles in promoting recurrence. It's a survival niche for surviving tumor cells. It is potentiating and promoting advanced recurrence upon tumor rebound, and that inhibiting this fibrotic response was successful in improving the efficacy of this immunotherapy. So moving forward, we're very interested in exploring how this could be adapted to other modes of treatment, such as radiotherapy, surgical resection, other kinds of immunotherapy, and then hopefully to move that into clinical application. So just to summarize now, so we took this very, I think, large-scale spatial multiomic approach where we integrated together single-cell RNA-seq, spatial transcriptomics, mass spec proteomics, and spatial proteomics, and then use this to explore this fibrotic response in the post-treatment environment of glioblastomas. And again, importantly, it wasn't only in response to this drug. We also saw this in radiotherapy, surgical tumor resection, other cancer models, and we saw very similar responses in patient samples. So using this spatial multiomic data set, we were able to identify how the fibrotic niche was potentiating recurrence, identifying that it was promoting a dormant stem-like state in surviving tumor cells. It was excluding phagocytic macrophages, and was also highly associated with T cell exhaustion. And we could also see that it was strongly promoting recurrence through the expression of a huge range of wound healing growth factors. And then finally, and I will stress at how critical the full spatial multiomic data set was in identifying the cellular mediator of this fibrotic response and figuring out which signals were actually driving this response so that we could design an inhibition strategy which was able to block fibrosis following treatment, and that this had the expected survival improvement for anti-CSF1R treatment. So with that, I would just like to thank the huge team that was involved in this project. So as you can imagine, with all these different multimodal data sets, there was a lot of really hardworking, talented people involved. Chief among them was Professor Joanna Joyce at the University of Lausanne. It's in her lab that all these studies were done. So she was absolutely critical to the success of this. I would also like to thank Anouk Zomer. She was my partner on this project. She was the co-first author on this cancer cell paper that you can see here. So if you'd like to read more about it, you can check it out there. I would also like to thank all of our very generous funding agencies. And also thanks to all of you for tuning in today, and again to 10X for sponsoring this webinar today. And if I'm not wrong, I believe I may have spoken very fast, so I think we have plenty of time for questions if you have any.
We definitely have time for questions. So thank you, Spencer. As a reminder to all of our webinar participants, if you have a question, feel free to type it into the Q&A box in the control panel. We'd also like to ask attendees to take a minute after the webinar has ended to take our exit survey to give us your feedback. So let's get started. The first question is, how do you account for immunogenicity of viral induction of tumor and your fluorescent protein onto TME?
No, this was definitely a challenge early on in developing this. So we tried various different background models, because these were initially in a mixed background. and we saw that that wasn't as much of an issue. We moved into the purebred Black 6 background, and then we essentially just had to do a lot of validation work to ensure that we were getting the same cell populations, the same growth dynamics, so that we could say with some certainty that we're not seeing a huge amount of immunogenicity against the GFP.
Okay. Does the fibrotic scar reveal any signatures of a hypoxic microenvironment?
Oh, absolutely. Yeah, the scar itself, especially the dense fibrotic scar, is massively hypoxic. We've seen a lot of really interesting what it does to vasculature that does, manage to make it into the scar is very dysregulated, is not, and then, looking at HIF1 alpha or anything like this, all the hypoxic markers are very highly expressed within the fibrotic part of the scar. And weirdly, this happens fairly early in the formation of these scars.
Okay, next question. How did you annotate the cell types in your Xenium data? Did you therefore integrate the data from your single cell samples?
In the Xenium data, we took a few different approaches. So this was largely done, with matching to databases and things like this. But we found that this wasn't successful for all the cell types. T cells in particular were very challenging. So because we had this limited gene panel, we didn't have a full whole exome within, because the question was about the spatial, right?
Mm-hmm.
Okay. Yeah, so because we were somewhat limited in the amount of add-on genes that we could include at this stage, we had to develop a certain strategy for some cell types that we could just take a broad, let's match it up to a database and it fits. But for T cells and I think other like lower propensity cells, we had to develop a specific gene signature of sometimes just like 5 to 10 genes to match them up to be really accurate. It also helped that because this was the mouse brain gene atlas or gene panel, and it was heavily focused on neuronal genes. So by excluding those from the cluster analysis, we got much better separation of the clusters.
Okay. Let's see. What is the origin of the stem-like cancer cells protected by the fibrosis? Is the initial treatment too conservative?
Like the absolute origin of those, I think, is something that we're not super sure of. I mean, if there is a cellular population that's already in some amount of fibrosis, like maybe within an oncostream or in the perivascular niche, we haven't really seen that it's these cell types. It could just be that any cells that were not actively proliferating at the time of treatment, or maybe they were spatially removed from where the macrophages are. Yeah, that's a difficult question to answer.
Mm. Let's see. What did you use for cell segmentation so that you could match up the spatial proteomics and transcriptomics?
So for this, we had our own machine learning model that we had developed through multiple centers as part of the Sierra UK Cancer Grand Challenges project. And so we basically developed a Stardust model that was based on mouse tumors across many different centers, and had a lot of different imaging experts annotate the cells to get a really good, accurate cell segmentation method. And then when we did the hi-fi analysis after Xenium, we ran this machine learning model on that. And then through Xenium Ranger, we were able to port those cellular segmentations back onto the data. And so I think because mouse samples can be challenging in terms of segmentation, because DAPI's very punctate and very messy, this was very successful for us.
Okay, great. And then dexamethasone has been linked to immune suppression in other contexts. So how do you explain it helping immune response in this model?
I think in two different ways. So in one, we're using it very short term. So this is, keeping it as short as we possibly can to achieve an effect, and not a very high dose. But also this is in combination with an agent that's specifically targeting the macrophage population. So in other cases where you're using dexamethasone in the clinic, these patients aren't always getting a macrophage targeted immunotherapy.
Okay. What do you think underlies the conversion of hypoxic cells from the scar into proliferating tumor cells?
Oh, no, that is a very good question and a very active area of exploration. This is something that we're working on now. It's very difficult to answer in the in vivo model. We tried different in vivo imaging approaches, but to do this with any number of mice, it was very much a needle in the haystack. So we're looking at approaching this with bioengineered 3D printed ECM in vitro so that we could be very defined and look at gene expression changes as cells escape from a dormant environment into a growth promoting environment. So that's hopefully, yeah, coming soon.
Okay. I think that might be all of our questions. Okay. Okay. So if there's no more questions, that's all the time we have for today. So we'd like to thank Spencer Watson and our sponsor, 10x Genomics. If we didn't have time to get to your question, or if you submit it late, we'll try to follow up with Spencer or other experts. As a reminder, please look out for the survey after you log out to provide your feedback. If you missed any part of this webinar or would like to listen to it again, an archived version will be emailed to all of the attendees. And thank you for joining us for this GenomeWeb webinar.
Thanks, everyone.
All right, we are